Okay, so thank you again, Nikhil and Madhuri, for that great set of talks. I'm going to try to build on this foundation and talk a little more about where CAR T cells may be headed in the future. So, so as far as the patients with multiple myeloma go, we know that CAR T cells today are being used, as Dr. Munshi just said, in patients who literally have run out of all treatment options. And there, as you saw, we are seeing some fairly spectacular responses with patients responding in excess of a year, some going out 2 years or more. But it seems like we're not seeing too many potential cures at this time. Perhaps there will be some patients that will have very long-term success and possibly cures, but it is hoped that in the future we could do better with CAR T cells, as Dr. Munshi alluded to, by moving this into earlier lines of treatment. So with that having been said, let me see here. Okay, so I'm going to move these slides here. Oh, sorry. Yeah, so we know already that there are clinical trials ongoing where CAR T cells are being moved into a line of therapy that is not the end line of therapy, but more into patients who have had 1 to 2 to 3 prior lines of therapy. It is expected that the CAR T cells will have better results when given in this less heavily pretreated population. Another thing that Dr. Munshi alluded to was perhaps moving these into even earlier lines of treatment, and for patients who perhaps relapse early after transplant. We saw a discussion of such a patient in the morning session, and we were all talking about what is the best treatment for these patients. And we certainly don't know the best answer. But CAR T cells could fill this niche. So there are already trials that are ongoing where patients who progress within a year of CAR T cell therapy, of transplant, are being offered CAR T, or even patients who progress within a year of standard therapy are being offered CAR T cells. The other thing, as Dr. Munshi already mentioned, is to see if we can use CAR T cells to deepen the responses. So we have patients who go through transplant but don't enter a complete remission. The question is whether giving CAR T cells to these patients to engender a complete response, possibly MRD negative disease, could lead to better outcomes. And once again, clinical trials are already accruing where patients fitting this demographic are being offered CAR T cells. The other is to do CAR T cells in tandem with transplant. There are moves afoot through the CTN, which is the transplant group, cooperative group in the United States, wherein CAR T cells are being planned to be given immediately, that is within a few weeks after transplant, to see if using both modalities of therapy together, followed by maintenance, will lead to better outcomes. And then people want to see if replacing transplant with CAR T cells is an option as well. The good thing is that CAR T cells may actually be even applicable and tolerable for patients who may not be candidates for transplant in a select few patients. So that opens up increasing options not only for the transplant eligible group, but for the transplant ineligible group as well. There is also theoretical reasons why we should use CAR T cells in earlier lines of therapy. Not only does any treatment work better when used earlier in treatment paradigm, but the T cells, which are the immune cells, are also thought to get damaged with multiple lines of treatment. And it is felt that the best quality of the T cells to produce the CAR T may be in an earlier line of treatment when patients have not seen too much in the way of drugs that can damage the T cells. And then obviously we want to see if we can cure patients with CAR T cells, and that obviously is the holy grail. If used earlier in lines of treatment, can we do that and actually eliminate the issue of progression? Now, some of the key scientific questions that are being asked today are why do T cells stop working? Patients have great responses, they enter MRD negativity, but often these are short lived responses. And there are a number of postulates that have emerged as to why CAR T cells stop working. On the cancer cell is eliminated. Of CAR T cells is targeting, is it possible that cells are losing BCMA? Or not only do cells lose BCMA, what we know is that cancer cells have a very different architecture. In some cells, perhaps even in the beginning, lack BCMA, and perhaps the uh, CAR T cell kills off the BCMA containing cells, and the minority of cells that are BCMA negative take over. There isn't much data to support this, but theoretically, that is possible. The other is that the T cells, just after time, get exhausted. They just stop working because of changes that these T cells undergo. And then there's the question of, are the T cells dead? Are they no longer even detectable? We know that in most of these studies, the T cells are persistent for several weeks and often a month, several months to a year or more. But in others, the T cells are not even detected after a few weeks. We do not know at this time whether the cells need to persist for long-term benefit or whether they can do their job quickly and disappear, and it doesn't matter whether they persist or not, and whether those patients where we don't detect them, are they just below the level of our detection, but they're still there? So these are questions that we need to answer to find out why CAR T cells stop working. Now, this is the structure of a car. My colleagues before have gone over this, but just to say that a CAR or a chimeric antigen receptor is a genetically engineered structure wherein you have this CAR construct, as we say, which has a number of different parts. This is the end, which is the T cell. So, and this is the membrane of the T cell, and this is outside the T cell. And what we know is that each one of these parts has distinct functions and can actually be improved upon, hopefully, in the future. So the external part of the CAR T cell is what recognizes the antigen on the cancer cell, for example, BCMA. So this part will recognize the BCMA. But then there is what we call a spacer, there is a linker, and then there is the structure of the CAR inside of the T cell that has different what we call domains. And this is a vital part of the structure that signals the T cells to expand and do its job. And we know that we have had success and failures in the structure of CAR T cell development, and that each one of these parts of the CAR is something that we can still improve upon. We can certainly develop CAR T cells that recognize the antigen better. So that is something that we know can happen by just having perhaps two hooks to the cancer cell. For example, the CARtitude study that Dr. Munshi just reviewed, that CAR has two hooks that it recognizes BCMA at two different points. So perhaps having two hooks is better. People are looking at this structure being fully humanized. Uh, Currently, the CAR T cells are using these FCVs that are not completely human. Some of them have murine elements that are derived from mouse antibodies. And so when you have that, the body recognizes the mouse antibody part of the structure as foreign and tries to eliminate it. So perhaps that could be improved upon. The spacer makes a difference. This has been also shown to possibly lead to side effects and loss of efficacy. And then real great emphasis is also being laid upon on improving the intracytoplasmic, that is the part of the CAR T cell, CAR structure within the T cell. Various co-stimulatory domains, as has been mentioned, are being tried. Co-stimulatory domains are the part of the structure that really tells the T cell that the brakes are off. You need to expand. And we have two commercialized versions of these uh, co-stimulatory molecules that take the brake off the T cells. One is called CD3 and one is called 41BB. One is called CD22. And so that also has differences. It is felt that some of these lead to more rapid proliferation, and in some there is more persistence. So those may also play a role as we design these CAR T cells. So we can certainly make those structures better. And as Madhuri said, we can actually try to go after new targets on the cancer cells. SLAMF7 or CS1 is the target for a drug that is already FDA approved, elotuzumab. CD38, which is the target of an antibody daratumumab and Sarclisa. Madhuri talked about excitement around GPRC5D as a target for CAR T cell development. Others include FCRH5 and still others. Now, what can we do to actually maximize what we already have? Can we try to make the CAR T cells that are currently in trials and close to commercialization work better? One way is to combine two CARs. The other is to try drugs that actually keep the target on the cancer cell so that the CAR T cells can go after them more effectively. What is known is that the BCMA, which is on the cancer cell that the CAR T cells recognize, is actually also actively being shed into the blood. And so when you have patients with high disease burden, they actually have a lot of this BCMA freely floating around in the blood. And what happens is that in a lot of cases, when you're giving a BCMA-directed therapy, the therapy gets soaked up by the circulating BCMA and may not be sufficient number of drug molecules to actually attack the BCMA on the cancer cell. People are talking about, so Notch inhibitors are now in clinical trials, and intriguing data is starting to emerge that these drugs keep BCMA on the cancer cell and perhaps could improve the efficacy of our CAR T cells. Checkpoint inhibitors are these drugs that have been used in solid tumors with great success. These again are drugs that are being combined now with CAR T cells to make them more effective by, again, taking the brake off the cancer cells, the T cells more effectively. Combining drugs that we use today, like Revlimid and pomalidomide, which are immune modulatory drugs, which rev up the immune system, they actually rev up the T cells. They could theoretically rev up the CAR T cells. So that is another way. The downside of some of these is that you could actually rev the CAR T cells up too much, and then you could actually unfortunately lead to side effects with these drugs. So you have to be careful. These have to be done in the context of a clinical trial. They can't be just done blindly. And then there are drugs like this class of PI3 kinase inhibitors, of which there are marketed drugs, which are thought to improve the ability of these CAR constructs to get into the more what we call memory T cells. So not all T cells are alike. It is felt that if you get into these more long-lasting T cells called memory T cells, you actually may have a better product. And so there are strategies that are being looked at to get these CAR constructs into the memory T cells. We have talked about some of the strategies shown here to improve upon CAR T cells. So we talked about trying to improve the antigen recognition by having two hooks onto the cancer. We talked about strategies to improve CAR T cell persistence, and some of these strategies are being looked at with combinations of drugs to improve the persistence of these cells we've already discussed. And then strategies to improve the targeting of the CAR T cells into the tumor using chemical molecules that these CAR T cells express is another strategy. And then, as we said, there are strategies to see if we can limit exhaustion and other reasons why CAR T cell may stop functioning. And Madhuri also talked about the fact that we are also looking at non-viral ways to transduce these T cells. That is using strategies called transposons, sleeping beauty. These are things that are too difficult to explain, but suffice to say that the CAR T cells being created today are using viruses to make the CAR enter the T cell in its production. And it is, though these have proven safe, people feel that going to non-viral strategies may be safer still and perhaps more effective. And there are, as Madhuri also stated before, other cells that can be used for CAR delivery. We are talking about T cells delivering CARs, so hence CAR T cells, but there are NK cells, which are another immune cell type that we are increasingly recognizing has anti-cancer properties. Those can be used to create CAR cells. There are invariant NK cells, so those are being used to create invariant CARs. And then there is this whole field of adoptive T cells that also is being looked at. This is a strategy that is able to go after not only the molecules that the cancer cell expresses on the surface of the cancer cell, but also molecules that are within the cancer cell. And again, I won't go into great detail, but just to say that the CAR T strategy can only go after antigens that are present on the exterior of the cancer cell. But there are a lot of antigens inside the cancer cell that could also be effective to go and develop T cell strategies against. And so adoptive T cells are being used in clinical trials as well. One of the other things that in the future may happen is what we call on-site manufacturing. The CAR T cells right now are, as you heard, being collected. You do the pheresis for the T cell, and these are shipped. It takes anywhere from, in the quickest circumstances, just over 2 weeks to sometimes 4 to 6 weeks for the CAR T cells to be manufactured at a central facility run by a pharmaceutical company. And these are obviously very expensive products. The price of the currently marketed CAR T cells for lymphomas and leukemias is in the $350,000 to $450,000 range for a single product. If these cells can be manufactured in larger academic centers, the cost of production of these cells is about $50,000 only. So you could get the product made cheaper, and you could get it more quickly possibly as well. But the fact is this does require literally industry grade manufacturing facilities to be present at the academic centers, which larger academic centers do have. Another thing that is also being looked at is what we call off-the-shelf CAR T cell therapy. So the CAR T cells that are currently being looked at and talked about so far are these autologous CAR T cells. Autologous means your own T cells are used to produce the CAR cells. So these require these complex logistics of manufacturing. There's delays, and we have talked about the current high cost of these as well. Whereas, one can actually give CAR T cells potentially in the future as an off-the-shelf drug that you wouldn't have to wait 2 to 4 weeks to get the product, that you could actually have these lying in vials in a pharmacy, literally, and that they're produced using normal, healthy donor-derived CAR T cells. There are certainly challenges there because when you use somebody else's T cells to produce CAR T cells, your body recognizes those CAR T cells as foreign. The CAR T cells recognize the recipient's body is foreign, and we face this tussle in allogeneic transplantation every day. So you have the threat of rejecting the CAR T cells if your body is overactive and rejects these. And then there's the other hand, there is the threat of the immune cells attacking the body and causing graft versus host disease. And so these things have to be worked out, but suffice to say, clinical trials with allogeneic CAR T cells have started. And then we talked about the site of delivery, which also is not a trivial issue. I think that patients need to be aware of this because they are their best advocates for change. Currently, and this is something that is sad to discuss, but is important to discuss. Currently, CAR T cells, as we said, are the ones that are commercialized are priced at a price range of $350,000 to $450,000. Now, the thing is that when you have patients who have private insurance, the private insurance usually covers the cost of the CAR T. But in patients who have Medicare and Medicaid, currently the insurances, the Medicare and Medicaid, doesn't even cover the cost of the CAR T. Instead of $350,000 to $450,000, the Medicare and Medicaid pays the hospital only a flat rate of like $280,000. And so currently the commercialized CAR Ts, most institutions, what they're doing is that they're making some money and saving some money off private insurance and subsidizing Medicare and Medicaid patients. In myeloma, where 2/3 of patients have Medicare or Medicaid, that kind of model may not fly. And so we have to be aware that it is very important to also look at how to make these things financially viable, because if these are not financially viable for institutions, a lot of institutions, despite the great results, may not actually offer these to patients, which is something that would be very sad. But the one way is to obviously change Medicare and Medicaid guidelines and say that we are going to pay for these products inpatient stay. But the fact is that that is often not the case. Now, one way around that, which is very practical and important to know, is to give the CAR T cells as an outpatient. Because of the unique structure of Medicare and Medicaid payments, when you give CAR T cells as an outpatient, not only can you charge the full price of the drug, you can actually charge the federal government 4% over the price of the drug, and then you are financially stable. But the fact is that these patients, if they are given CAR T cells as an outpatient, first of all, you need to have the infrastructure, you need to have the ability to deliver them as an outpatient, but also what is important is that you need to keep patients out of the hospital for 72 hours after the administration for the federal government to pay you the full price of the CAR T, which means that if patients develop CRS within 24 to 48 hours, it will be something that will be very difficult to do. There are certain CAR T cells that are having CRS emerge at 7 days. Those may be more viable for outpatient use. So these are practical considerations for the future that also are important to know. So thank you very much, and that's all I wanted to say.